start off with you, so over to you. And Janet, you are muted, by the way.
Great. So hopefully you can see that. So it's lovely to see so many of you this evening. We're going to talk about the Breed for Change project, which a lot of you will have heard about before and know that we're involved in, all about trying to breed sheep with a lighter carbon footprint. So I'm going to start off talking a little bit about what the project's all about. That will be familiar to a number of you that have heard me talk about it before, but new to some of you. I'm going to give you a bit of an update on progress, and then I'm going to hand over to Joana to do a much deeper delve into breeding for the microbiome, which is part of this project and a part of it that both Dewi and I find increasingly fascinating. And Joana is a great specialist in that area and has some very interesting insights into it from her work with cattle. So hopefully you'll all enjoy that as much as I know I will. So what's Breed for Change all about? Well, Breed for Change is a project that Innovis are leading, but we have lots of partners in it. We're doing it with other breeding groups of maternal sheep, so the Exlana breeders, Performance Recorded Clin breeders, and the Centurion Dorset group, as well as lots of industry partners who are supporting us, and academic partners, including SRUC, and Joana works for SRUC, and Harper Adams. And overall, basically our aim with it is to try and find the best breeding strategy to sustainably reduce methane per kilogram output from ewe flocks. It's funded by DEFRA through their Farming Innovation Programme for their small R&D project, and it runs from June 2023 to May 2026. So we're about halfway through on the project. And really, we're kind of taking three parts at trying to this breeding strategy. We're looking at ewe efficiency, something that you'll know from an Innovis point of view, and the other breeding groups is something we've been aiming to improve over a number of years and have had quite a lot of success in doing that. We're looking at health of our lambs, because that's obviously important for sustainability. And we're looking at the methane emissions. So why are we doing it? And this is an argument that's been reiterated a number of times over recent weeks with the controversy over the ALA and also even Hilda, the SRUC news from earlier in this week. So why are we looking at trying to breed sheep with a lower carbon footprint when sheep farming isn't the problem? And when we look at sheep farming in the context of other emissions within the UK, so if you ignore these bottom two at the bottom for a minute, but look at the others, agriculture is sitting there not as the highest emitters by any means, but having quite significant emitters because we are quite a big industry. If we then divide the agriculture bit down, the majority of that can be attributed to livestock, and a small proportion of that livestock can be attributed to sheep. But it's very, very small compared to many of the other sources. So sheep aren't what's causing climate change. And equally, breeding sheep with a lower carbon footprint aren't going to suddenly reverse climate change. And I think it's important that we keep that in perspective. So why are we doing this? Well, we're doing it because with a problem like climate change, and I'm sure many of you will agree with me on this, I think we all need to do our bit. Individually, none of us are going to be able to have a big impact, but if we all do the best we can within what we can influence and what we do, collectively, globally, we will have some impact. So it's part of doing our bit. It's also, for sheep farming in the UK, a very important part of looking after our markets. The retailers, the processors are all very keen to reduce their carbon footprints. And a lot of their carbon footprints can be attributed to before the farm gate. So they're looking for product that has a lower carbon footprint. Consumers are being told, rightly or wrongly, but they're being told that one of the easiest ways for them to reduce their personal carbon footprint is to reduce red meat consumption. Now, obviously, we don't want them to do that, but we want them to be able to access red meat and lamb where they feel that it has an acceptable carbon footprint. Our competitors are already up in this game. We look at Ireland, New Zealand, etc., competing into our market. So looking after our market is a very important part of why we think we need to do this. And then there's the government drivers as well. They've set clear targets for net zero by 2050. And although they haven't set specific targets for agriculture, agriculture is expected to play its part. And we can be almost sure that if we don't play our part as sheep farmers in starting to reduce our carbon footprints, it will be imposed on us one way or another. So it's about taking control and doing our bit, looking after our markets. Why methane? And again, many of you will know this, but when we look at that contribution from livestock and the contribution from sheep production, the largest portion of those emissions come from methane produced from the natural process of digesting grass and forages. So that's the obvious low-hanging fruit to aim for. And if we look at the flock and where that's coming from, it's the ewes that are in the flock 365 days a year producing methane that are the largest contributor to the flock emissions. So our target is to reduce methane emissions from the ewe flock because that's the main source. Now, something that's important is because we're not going to change climate change through breeding sheep with a lower carbon footprint, if we're going to do it, everything we do has to be a win-win. It also has to benefit the overall sustainability of our sheep flocks. And we know that sheep that contribute to sustainable reductions in emissions need to produce high quality product efficiently. They will have a lower carbon footprint, and that's a win-win because it's going to be better financial returns for the sheep farmer as well. They need to optimize the use of grass and forage crops because that will reduce input costs. It will increase carbon sequestration. It'll improve soil health. And it means we're producing protein from less productive land. Lots of win-wins there. They need to be healthy and therefore productive. That means they'll increase output. It'll also reduce input costs. It reduces drug use. And most importantly, that will also improve farmer and sheep well-being. So again, lots of win-wins in terms of overall sustainability. And they need to produce less methane per kilogram of feed that they eat by digesting that feed more efficiently and producing less methane from it. So they reduce direct methane emissions. It's estimated that about 12% of feed energy is lost through the production of methane. So if we can get them digesting that feed more efficiently and producing less methane, we get more from the feed they eat, another win-win. And we know that sheep integrated into arable systems can have a very positive impact on soil health. So when we're thinking about breeding sheep that are going to contribute to sustainable reductions, we need them also to be attractive to integrate into arable systems, so easy to manage on forage. So we bear all those things in mind in the project in terms of trying to develop breeding tools for lower methane per kilograms output. And if we think of the things that are driving that methane and how easily we can breed from those, we've got a number of cogs in our machine. Ultimately, we want to reduce the amount of methane per kilogram of output. So we can do that by increasing the kilograms of carcass produced per ewe. Now, that's something that we've been doing in our breeding strategies for many years. And we've seen very significant progress in all the lines that we're working with in terms of those traits. We have EBVs that are associated with output, and we're used to using them. So things like lambs reared, maternal ability, lamb growth, etc. So in this area, this part of our machine, we're quite confident with it. Another important cog is going to be the dry matter intake of the flock, because our methane outputs are highly related to the amount of dry matter intake. So EBVs that are associated with traits influencing the feed required, ideally, we want to be able to drive that dry matter intake per unit of output down. So those are traits like ewe weights and body condition scores, longevity, health, because that influences days on farm, feed efficiency, etc. And again, these are traits that we have been working with over a number of years, but we can improve what we do. So I would say they're a work in progress. And part of the Breed for Change project is developing those traits further, getting more data, so we have better breeding values and better breeding strategies to help in that part of our equation. And then the novel part for us is the methane cog in here. So if we can also, reduce the methane produced per kilogram of feed eaten, that will also help us breed for our lower carbon footprint. And that's a new area for us. So that's something that we're putting a lot of work into, collecting data, understanding how that interacts with the other traits, and working out how we build that into our breeding strategies. So in order to do that, we need to collect a lot of data. And so the kind of first part of the project is collecting data to help develop the breeding tools we need to improve those different parts of that equation, and also building into that genomics so that we have genomic predictors giving us extra accuracy and being able to spread the benefits wider in the population. Now, as I said, dealing with that methane trait is a new area for us, and a lot of emphasis has gone into collecting data on that, which we're doing using the pack trailers, the portable accumulation chambers, and we've been collecting data on that for the last 2 years now, and you may have seen it traveling around the countryside. So to December just gone, we've now collected a lot of data in many of our flocks, over 7,000 records in total, over 7,000 sheep in total, building up a significant database on that so we can develop breeding values and understand relationships with other traits. And we have more to do in the next few months. We have about 500 Dorsets to measure in February and more of the Innovis lines to measure later in the spring. And then another year of measuring next year. So we'll end up with around 12 or 13,000 animals measured, feeding data into allowing us to answer some of the questions. So what have we learned from that so far? So we've analysed the data from 2023, which is about 3,000 animals measured. And we found out that there's an awful lot of variation out there. Typically, there's about a 40 to 50% difference in the amount of methane produced between the top 20% and the bottom 20% of the flock. So there's an awful lot of potential for reducing methane emissions. And this analysis here has been done by Karolina Kosija from SRUC, so one of Joana's colleagues who's analysed this data. And one of the first things that became very obvious to us looking at the data was there was a very strong relationship between the live weight of the lamb when we measured it and the methane per day that it was producing, a phenotypic correlation of about 0.75 to 0.8, which is very high. So immediately we know that just measuring the methane output per day and selecting on that probably isn't a very sensible approach, and that we need to do something more sophisticated, certainly in terms of breeding values, but also in terms of adjusting that data for live weight and other factors that will influence it. When we do that, we can look at the heritability of traits, and we found that the heritability of methane in terms of grams per day, when we adjust it for live weight, is at around 10%, 8 to 10%, which is quite low, but still something that we can change if we want to. And that estimate, although it might seem low, is actually very much in line with other estimates that have been found in other parts of the world when they look at this adjusted for live weight. Another trait that we're interested in is this bottom trait, which we call total gas ratio, the ratio of methane to methane plus carbon dioxide. And we find that, even after adjustments for live weight, to be quite a heritable trait, 0.23, which is kind of in the region of something like weaning weight, which we know we can change very readily by selection. So this is a trait that we're interested in because a lot of other studies have shown that it's very closely related to dry matter intake, or methane produced per kilogram of dry matter intake, which is really the trait that we're after, that efficiency of digestion that's producing less methane from the rumen. So we think this may well be our target trait, and we're doing a lot more work this year with the data we've collected to try and look at that trait in more detail and look to see if that is the trait that we actually move forward with in terms of breeding values as the trait that we want to select for in future. But it's something I can only emphasize from the work that we've done so far is that just using raw phenotypes is probably not a sensible thing to do when we're looking at methane measurements. In terms of next steps, Carolina's going to be analysing data again this next year. She's going to be integrating genotypes so we can get to the point of being able to produce genomic breeding values for the traits that we want to define, and to look at the relationship with those other traits that are important for production efficiency. So we really understand those relationships and we can define a sensible breeding strategy going forward. And of course, at the same time, methane is a new and exciting trait, but we're also looking at improving those other important cogs in our machine, improving efficiency and improving health. Now we have a fully performance recorded population of a lot of sheep to start with. So in terms of improving efficiency, we have a wealth of background data to draw upon in terms of that and to understand relationships of traits with that. Two areas we're putting extra work in to really get more data is measuring U weights and body condition scores at multiple points throughout the production cycle so that we can really understand how that changes and how that might be related to methane traits so that we can make sure that we're producing sheep that are going to thrive in our forage-based systems. And we have about 58,000 measures collected so far on those. So we're building up a really strong database on that area to be able to do some really good analysis of relationships and selection. And we're putting extra effort into looking at parasite resistance as one of the main limitations in lamb health by doing fecal egg count and IgA measurements on lambs individually to get a much better measure of resistance to nematodes. So we can really sharpen up our selection for that and again understand relationships with overall efficiency and our methane traits. And over last year and this autumn, we have over 5,000 plus records collected so far across all the breeding groups. So we're developing really deep data sets in all these areas to be able to sharpen up our selection and improvement. Something else that we're very keen to do is to really understand if we're going to select sheep that are more efficient and that produce less methane and digest their feed more efficiently, what are we actually changing? And are those sensible things to change? We want to understand the biology of what we're doing better so that we can avoid unintended consequences and we can make sensible decisions about the breeding strategy. So we have a subset of lambs on which we're making much more detailed measurements and data. They're mainly in the Innovis Southfield nucleus flock, and the work that Hamish, Leah and Kate are doing there to record data in that is absolutely invaluable to this project, but also more detailed measurements being made in the Harper Adams Clin flock as well, so we can look at a variety of breeds. So at Southfield, we're not only measuring methane in lambs, but we're following those lambs throughout their lifetime for the duration of the project. So we can understand how that might change over their lifetime. Now, work that's been done in New Zealand and Australia has certainly shown us that when we rank animals on their methane emissions as lambs, their ranking stays pretty similar over their entire lifetime. But we want to check that. And we want to check that against our other measures of efficiency. So we've got 4 cohorts of animals that we'll be following through their lifetimes to get those measurements. As I've said before, we're monitoring body condition throughout the lifetime and the production stages of the sheep, so we can really understand, are there differences in body condition of the low emitting sheep compared to the high emitting sheep? Because monitoring body condition is the best way of finding out that sheep are thriving in our systems and how efficient the ewes are in terms of harvesting the feed that's available for them and turning it into body condition. We're also making detailed measurements of feed intake and efficiency. And I'll talk a little bit more about that. So we're doing that in the Southfield lambs, roughly 250 a year, and also Harper Adams are doing that with their Clin lambs. For the Southfield lambs that are going through our feed efficiency unit, we're measuring their rumen volume through CT scanning. We're sending them to the CT scanner in Edinburgh and getting a measure of their rumen volume. And again, I'll talk a little bit more about that. Then the really exciting bit, which Joanna's going to talk about, we're monitoring the rumen microbiome. So all those lambs that are measured for feed efficiency in CT are having a rumen sample taken. And because we're seeing real potential for that data, we're extending that number to more than that. So we get a bigger data set on that to look at. Of course, ultimately, we want all our sheep to graze, and grazing intake and behavior is really, really important. That's really hard to measure and something that's probably beyond the scope of this project, but really important. And so Harper Adams are doing some pilot work to look at ways of measuring grazing intake and behavior and to relate it to their measures of feed intake and efficiency that are done in pen sheep on grass nuts, to start to establish relationships there and work out how we can look at that more deeply. So looking at some of these things in a little bit more detail. So measuring feed intake and efficiency. So far, we've put 3 batches of 125 Innovis lambs through a feed efficiency unit, which we host at Southfield, our nucleus flock near Hoyk. And we have automatic feed equipment for that on loan from SRUC. And you can see a picture of it here. And 120 clins have been put through the Harper unit, where they don't have the benefit of automatic feeders. They've got lots of students, weigh in buckets, but also get very accurate measurements. The general principle is that these sheep, which remember are fully performance and recorded on grass, so we know how they perform on grass before, they come into the feed efficiency unit. They have 2 weeks of training to learn to eat grass nuts. And believe me, it takes 2 weeks to train some of these to learn to eat. And then we have a 6-week measurement period where we're measuring individual feed intakes of them every time they go to the feeder. We're feeding ad lib grass nuts to them, and we take weekly weights. They have an ultrasound scan at the start of their measurement trial and a CT scan at the end. Now, the reason for that is so that not only can we estimate intake and change in weight, but also change in body composition. But the CT scan at the end gives us that added benefit that we can also measure the rumen volume of them. So we've had 3 batches through so far. We have another batch starting next week of ewe lambs at Southfield, 125 of them. And then we'll do another 2 batches at Southfield in 2020, the back end of 2025 and beginning of 2026. And the ones that are done at Southfield, so the ones that we did in the autumn this year and the ones that will be done, the next 3 batches that we'll do, also have a measurement of methane in the pack chambers done towards the end of their feed intake trial. So we have a very direct measurement of their methane production while we also have accurate measures of feed intake. Now, one of the aims is so that we can start really narrowing down on that trait of methane produced per kilogram of dry matter intake, which is the trait that we're really interested in. As I said, in order to try and relate intakes in this rather artificial system for us who are used to seeing grazing sheep to how animals graze, Harper are going to do some grazing trials at the later end of this year in 2026 to look at those relationships. It's probably at the pilot level in terms of the number of sheep they're going to be able to monitor, but it will give us some interesting insights into that. Certainly a lot of evidence in other countries is that although it is quite different behaviour when sheep are housed like this, their intakes and their efficiencies relate quite strongly to how they do at grazing. So hopefully it will confirm that. So we're also, as part of that, delving into the biology a little bit deeper, measuring the reticular rumen volume of those lambs that are going through the feed efficiency unit at Southfield. 487 lambs have been measured so far, which is a little bit more than we've measured through the unit, but that's because we've also scanned some ewe lambs that were born in 2022 at the beginning of the project to up our numbers a little bit, and because we wanted to follow them through their lifetime. They go through the CT scanner at Edinburgh. Kirsty and John do a fantastic job there. And it sounds very easy, doesn't it? You put them through the CT scanner and you get a measure of rumen volume. Actually making that measure of rumen volume requires a lot of work, and we're very grateful to the work they've put in developing the algorithms to do that and being able to do it. But we're getting that measure of rumen volume. So these are sheep that will all be managed identically. They're coming off the feed efficiency trial, so they've been fed identically. We've got measures of feed intake, and then we're measuring their rumen volume through the CT scanner. We've also taken a microbiome sample from 245 of those lambs that have been analysed so far, and Joanna will tell you a little bit about that. And a further 375 will be measured in the next 2 years to build up a bigger data set on those. So really interesting data coming out of those measurements. One thing that always amazes me, given that these sheep will be measured, treated identically, they've been fed on the feed efficiency unit. Average rumen volume is about 7 litres, which is what we'd expect for a lamb of that live weight. But even if we adjust that for live weight, take those live weight differences into account, we're getting a massive range of rumen volumes from about 4 to 9 litres. So huge amount of variation in the rumen volume of animals. What have we learned so far from that feed efficiency and rumen volume work? Well, it's very early days from this, but Nicola Lamb at SRUC has been analysing the data. And as I said, it is early days and there's much more interesting stuff going to come out of this. But we're seeing a lot of variation in rumen volume. We're also seeing a lot of variation in residual feed intake, which is the way we measure efficiency as being the amount of feed an animal requires to achieve a certain level of growth. And we're seeing differences between our lines in dry matter intake for the same performance of up to about 0.4 kilos a day. So quite big differences. And we're seeing consistency across the different groups we're doing and the different years as well. So there's some really interesting things coming out of that. There's also a suggestion that there's a slight relationship, not a strong relationship, but a slight relationship that larger rumens are related to higher methane outputs after we've adjusted both those things for live weight. It's not a strong relationship, but there does seem to be a tendency, which would agree with other work that's been found in Australia and New Zealand. And there's a plot here. We've got the ewe lambs and ram lambs are the two different colours, which is why you see those. And you can see a very slight tendency with rumen volume along the bottom here. You can see ranging from 5 up to about 10. And the methane going up the side. A lot of variation around it. So there's certainly outliers in there, but a very slight tendency. So if we selected for lower methane and ignored rumen volume, we might reduce it. But if we understand that relationship, we know there's plenty of variation there where we can select animals that have lower methane, but still have a adequate rumen volume. It's not a necessary reduction that we would see. But the important thing is that we understand it and we have that data to follow it. So that's all about trying to develop the breeding tools we need and knowing how to use them sensibly. So we're talking about EBVs, genomic breeding values, and indexes for those key traits. We're building up the data so that we can understand the genetic relationships between the traits, so we can build it into indexes and avoid unintended consequences in our selection. We can get the win-wins. As part of that work, we're looking at how genetic changes will influence methane per kilogram product in terms of our current indexes, so that we'll be able to tell you for Innovis lines where you've been already buying sheep, how much that might have already been expected to reduce your carbon footprint. And ultimately, we want to be able to produce an index that will allow us and you to rank animals on their breeding potential for reducing the carbon footprint of the product you produce, so that you have the option when buying rams to be able to identify the ones that are going to help you reduce the carbon footprint, both in terms of producing more efficient daughters and intrinsically digesting their feed a little bit more efficiently and producing less methane from it. So that's where we're going with it. I'm going to hand over now to Joana to telling us a bit more about breeding for the microbiome, which is a fascinating subject and something that we really see real potential for, both within this project and beyond. So I'm going to stop sharing mine and hand over to Joana.
Thank you. I'm just
Janet, whilst Joanna's pulling that up, there's just one question that be quite good just to cover, I think.
Okay.
John Martin, a good friend of ours in Ireland, has put a couple of questions up. One was actually asking how we measure feed efficiency, which I think you've covered to some degree, but also the question, is body condition score an indication of feed efficiency within a population in the same management group?
Okay, I think that's a great question. So we're measuring feed efficiency, which hopefully I have kind of answered in that we have those automatic feeders. We can measure intake quite accurately. And then we're also measuring changes in body weight and body composition. So we can see for how different sheep are eating different amounts of feed for a different level of growth. Is ewe condition a measure of feed efficiency? Well, I know others have argued, and I certainly would argue, that when you're measuring, you've got a group of ewes being measured the same way. Some are putting on condition, some are not putting on condition or not putting it on as easily. Why is some doing it more? Well, that maybe there are differences in the way they graze, but ultimately feed efficiency has got to be part of that, hasn't it? If a ewe is using her feed more efficiently, she's going to put on condition. So I think it is an indicator of feed efficiency. I think there are other indicators and we can build on, but that in itself is partly an indicator.
Thank you, Janet. And obviously, John, if you want more on that, just email me and we can get you in contact with Janet to have a better explanation. Just to finish on some of these points before we go on to Joanna. Bill Grayson, Janet is asking how the pack chambers work, how were the emissions from the sheep tested, monitored, and how long at a time? So the mechanics of the pack chamber, really.
Okay, so the way the pack chambers work is they're portable accumulation chambers, and it's all about accumulating gas, the gases that sheep expires while they're in there. So the lamb goes into the chamber for 50 minutes. We measure the gases in the chamber when it goes in, at which point methane is 0, carbon dioxide is 0, and oxygen is at about 16%. As it's in there, it uses oxygen, so oxygen goes down, it breathes out carbon dioxide and emits methane, so they increase. So it's literally measuring how they accumulate over that time period of 50 minutes, and then we multiply that up to work out grams per day. The important thing is that the sheep are all measured in a way that they're all managed identically beforehand, so they're grazed together on at least 1,500 kilograms dry matter grazing for 3 weeks beforehand. We don't take any measurements. We get them in. We don't take any measurements within the first hour, because when a sheep eats, it has a peak of methane production that then falls down, and we don't want to get any of those peaks. So we make sure everything can't have eaten for at least an hour before we measure them. And they mustn't have been off grass for more than 4 hours when they go into the chambers, so that we're getting quite a uniform time that we measure them over.
And just to expand on that, Bill, in terms of the time of the day, what happens is that there'll be group brought in the morning, they're weighed and split into groups of 12, because 12 sheep go into the trailer at any one time individually into the compartments. And then they're mixed side groups to make sure that there's ranking that can happen. And then 4 groups normally are put through in the morning. The afternoon sheep will have been put straight back out in the morning after being weighed, and they come back in then at lunchtime. They stay off feed for an hour, and then those 4 groups are then done in the afternoon. So they're done in batches of 12, normally 3 or 4 runs in the morning, 3 or 4 runs in the afternoon, but always after coming off feed for at least an hour, being on that feed for about 3 weeks, no changes, no shearing or management things that would have disrupted them, and all treated the same. And then the afternoon group will come in and be off feed for an hour, and then they'll be out before that magic, normally within a sort of 4 or 5 hours done. So hopefully that gives you an idea. But again, Bill, if you want any more info on that, please get hold of us, and we'll work on it. Yeah, so it's very much, as you say, a pre-test feed regime, done very much to get things as even as possible within all of that, really. So I think our friend Emily has put something up, Janet, which is what live weight does the 0.4 kilogram difference in daily dry matter intake apply to? So I suppose what Emily's asking there, Janet, is that 0.4 difference, although it's adjusted to weight, what's the sort of size of these sheep in that element?
So I think you're right. It is quite a significant difference. It's quite, yeah. The average weight of the lambs is probably about 45 kilos when they go in. And it's quite interesting, actually. I think last year we struggled a little bit with the feed efficiency trial in terms of training sheep to eat the feed and the quality of the grass nuts we were getting. So we didn't have fantastic live weights of some of our batches going through last year, but we found that sort of difference. So some lambs were still doing really well and others weren't doing so well. This year it's gone much more smoothly, better quality feed. We've kind of learned how to get them trained a little bit better, and we had much better intakes this year. And our ram lambs this year, we had an average growth rate of 200 grams a day, with some doing over 400 over the trial. But we still found the same sort of differences. and consistent as well. So I hope, yeah, it is a big difference. And if you think of it in terms of feed you would grow to maintain a certain level of production, it would make quite a significant difference at a flock level.
Emily, please, if there's any more you want to know, you know where we are, please get hold of us. We will put an email address in there for you afterwards, all in case you want any more information. So I think based on that, we will just move on to Joanne.
Okay. Can I just confirm that you can see my mouse?
Yes, your mouse is running up and down the hills.
Perfect. Okay. Okay. So hello everyone. My name is Joana. I'm a researcher at the SRUC in Scotland. Actually, this is my first project with sheep. My background is in beef cattle mostly, and more recently a little bit of dairy cattle, but everything it's microbiome. So I think that there are a lot of similarities between the microbiome within ruminants. And so I'm going to give you a little bit of overview of rumen microbiome work done in SRUC in the past few years, and then a little bit on the analysis that I've done in the on the sheep data. So I'm guessing that everyone knows this, but we will see. So the rumen microbiome is very important for the ruminant, and this is because the ruminant does not produce the necessary enzymes to digest the fibers that it eats. So the plants that the animal eats are full of fibers, and this is the main source of energy and protein for the animal, but the animal actually does not digest this. It has to rely on the rumen microbiome to gather nutrients from this fiber. And so in the rumen microbiome, we have several different microbes. Bacteria, protozoa, and fungi are the main digesters of this fiber. So they do the fermentation of this fiber, and while they do this fermentation, they release volatile fatty acids that are the main energy source for the animal. And they also multiply and grow. And in that process, then the microbes themselves follow through the digestive tract and become themselves the microbial protein source for the animal. And in the process, they also produce vitamins, which is very good, obviously, because it feeds into the animal growth. And in the case of the dairy cattle, it contributes for the milk yield. However, we have an issue with the rumen microbiome, or we see it as an issue. That is, we also have archaea organisms here. And the archaea organisms use the hydrogen and carbon dioxide that are produced during the fermentation. They use it for their own metabolism, and in their own lives, naturally produce methane. So this is the big issue. One of the things that Janet said, and is correct, that some of the energy that the animal is eating, instead of going for the volatile fatty acid production and microbial protein production, is going into releasing hydrogen and carbon dioxide, and it's going into the archaea and released in the form of methane. And so we are having a little bit of waste of what we are feeding to the animal because of this natural process. The other issue is that obviously it contributes to the environmental impact and the greenhouse gas emissions. And so ideally, we want to continue producing the animals, but reduce a little bit this methane here. And if possible, transform this energy that we are wasting here into growth or yield. And so one of the works that we've done in SRUC quite a few years ago, maybe 2017 or 2018, was on difference between diets. So in this case here, these were beef animals, beef cattle. And what we had here were animals that were fed either a grass silage or a red clover silage. And we saw that there were differences in terms of methane. So you can see that the grass silage fed animals were producing a little bit more methane per kilograms of dry matter intake than the animals fed red clover. And then in this paper, we looked at the composition of these diets, And we saw that the grass silage was richer in fiber and in sugars, and it had also a decreased passage rate, whereas the red clover was richer in pectins and richer in proteins, and it had an increased passage rate. What we also saw was that in terms of production of volatile fatty acids, which are then the energy source for the cow, the grass silage was also increased in relation to the red clover. So what we had was a higher level of fermentation associated with higher methane production. And this is sort of logical, because when we are having a little bit more fermentation, then we have more hydrogen and more carbon dioxide, and therefore the archaea are more active, and then they produce more methane. So we have, when we are talking about the microbiome and thinking about how we can make the microbiome produce a little bit less methane, we need to account for this kind of logic that happens in there, is that the more fermentation we will have, the more hydrogen, and then the more, very directly and very roughly, but still true. So the take-home message from this paper here, and over these papers, I will always have a little box here that tells you which paper it is. So you can find this online if you want to read it and go through all details. It has here the authors, and so it's very easy to find. So the take-home message is that different diets lead to differences in methane emissions, as expected, because microbiome is the main actor in terms of the fermentation and of the methane emissions. Then we had a very nice paper as well. So this one, we did correlation networks. And so this seems a little bit, maybe a little bit complex, but it's in fact very easy to understand. So these two here are the same network, but they are colored in different ways, just to give you two different kinds of information, but it's about the same thing. Each node here is either a microbial gene, or a microbe by itself, a bacteria, or an archaea, or a fungi, or a protozoa. And when I have here the plus in red, it's because, so we have this, sorry, let me just explain better. So the node is each one of these variables, the microbial gene or the microbes. And then when we have an edge between two nodes, it's because they are correlated with each other, which means that if one grows, the other one grows as well. And so we can make a little bit of connections between this, and we can create clusters. And that's what we have on this side here, that we have these clusters here in the legend, and we have different colors for different clusters. Here we can see the type of information that we are dealing with. So here we can we have here in the green microbial genes, we have the blue that are the bacteria, and we have the yellow that are fungi, and then a little bit scattered around are the archaea, and the black one are the protozoa. Here is a cluster that is made by a statistical method, and it just means that the entities that are in the same cluster are closer together than they are with the other that are in the other cluster. So they just have like more things in common. So the red plus here means that the cluster was associated with increased methane production. The green, it was a cluster that was related to a decreased methane production. And finally, well, we were expecting when we looked at this, when the authors actually produced this, because this is not my paper, it's a colleague's paper, when she looked at this, we were thinking that the clusters that were going to be the ones more related to methane, that they would have a little bit more archaea, because the archaea are the ones that produce the methane. And in fact, what we saw here was that when looking at each cluster and how much they account for the methane production, the one that was more related to the methane production is this green here that I have the arrow, is this green here. It was called the methanogenesis cluster, and it explained just the entities that are here, that if we look here, we can see that are microbial genes and a little bit of bacteria. It explained 57% of the methane variation in the animals. But then we looked the advantage of looking at microbial genes instead of looking at microbes by themselves. When we look at microbial genes, we can see the potential function of these communities. So we can see what are they doing there, not who they are, but what kind of capacities they have in the rumen. And so when we look at this what kind of functions these were doing, they were related to nitrogen fixation, lignin degradation, amino acid metabolism, and sugar metabolism. They were not related to the methane pathway. So this gave us a little bit more hint that the production of methane is not just this organism produce methane. It's a whole ecosystem that is working in a certain agreement between them, and sometimes disagreement, to then produce this methane. And one of the key aspects of this is the part of the fermentation, the production of the volatile fatty acids, production of hydrogen, and then who is going to use this hydrogen? Yes, the archaea will use it. But in fact, the most important thing is the profile of the fermentation. And so I've written here some little things that I think are important from this paper, but the take-home message from this one is that differences in methane production are more associated with fermentation patterns than actually to the methanogenic organisms. Okay, so this one I really like. This one was my first paper. And in here, we look again at beef cattle. And here I was looking at the association between microbial genes in the rumen in relation to performance traits. And I looked at 4 performance traits: feed conversion ratio, average yield gain, residual feed intake, and daily feed intake. And from 3,000 microbial genes that we saw in the rumen in these cows, in these steers, we tried to identify which ones were the most important and more closely associated with each one of the different traits that we were measuring. So we identified 20 genes here for the feed conversion ratio, and these 20 genes were associated with 63% of variation. So it explains a lot of the variation in feed conversion ratio. The same way for the average yield gain, so 14 genes for 65%, residual feed intake 17 genes for 65%, daily feed intake 18 genes for 73%. One of the things that we noticed, and that should be just by the way that we calculate the feed conversion ratio, is that the feed conversion ratio and the average yield gain were correlated with each other. So the higher the average yield gain, the lower the feed conversion ratio. So the correlation between them was -80. So it's quite strong, but it goes in opposite directions. The same thing, more or less, we observed in the residual feed intake. So the more residual feed intake we have is associated with more daily feed intake. The association was a 0.77, so it was very, very strong. And then no real correlations between the other pairs. A funny thing was that although from 3,000 genes, we identified only 20 and 14 in this case, here we had 6 in common. So there may be a microbiome basis for this correlation that we have between the traits. And the same way on the residual feed intake and daily feed intake side, that we only have 17 and 18 genes that we identified for prediction of each one of these things. And then we have high correlation between them. And the same way, we have a little bit of overlap of the genes that were associated with each one of the traits. And then again, we looked at what are these genes doing, what kind of functions are they able to do? And what we noticed was that the animals that had improved feed conversion ratio and improved average yield gain had increased abundance of genes related to cell wall biosynthesis, hemicellulose and cellulose degradation, so fiber degradation, and also host microbiome crosstalk, because there are a few interesting things about here. I don't want to go too much into this crosstalk, but I thought I would mention it just in case someone gets curious about it. In terms of residual feed intake and daily feed intake, we looked at the animals that had better residual feed intake and daily feed intake, had the microbial genes more associated with vitamin B12 biosynthesis, a little bit of environmental information processing, which means that the microbes probably are in the environment trying to understand if it's a good time to multiply and reproduce. Is it a good time to stop reproducing and maintain the numbers that they have at the moment, and also bacterial mobility. So the take-home message here is that the microbiome associated with performance is involved in very interesting microbial functions, and functions that we would, at the first look, we look at them, vitamin B12 and all of these things, and we can see why they could be related to increased performance in the animal. Then, now we are going for a little bit more genetics. So this is the work of Rainer Rowe He's SRUC as well, and he actually was my supervisor during the PhD. This work is again in beef cattle, and so what he did here was measuring methane and comparing sire progeny groups. So this work is from 2016, and we've advanced a little bit from this, but I still think that there are quite a few important things in here. So what we have, I think that the main conclusion from this paper is that the differences in methane emissions between sire progeny groups were larger than differences between diets. So we knew that we were going to get different methane when we feed different diets to the animals. The diets here were, one was forage and one was concentrate based. So we knew very well that since the fermentation is different, volatile fatty acid hydrogen production is different, archaea react in a different way, methane is going to be different. That's fine. What we didn't know at the time, or they didn't know at the time, was that the sire progeny groups differences were going to be so big. And this indicated obviously substantial genetic influence of the host animal over the microbiome. And if there is this kind of influence, then there is a possibility for breeding. They also went a little bit further. They looked at a little bit of microbiome, not in the detail that we are looking at the moment, but still looked a little bit at microbiome in terms of archaea and bacteria ratio. And they noticed that there was a direct genetic influence of the host on the rumen microbial methane production. And so take-home message here is that the host genetics does have an influence on the microbiome profiles. This is one of the most recent ones, and it is very, very close to, I would say, state-of-the-art. So in this case, we used, again, beef cattle. This beef cattle was measured for methane yield. The methane yield was, the heritability was calculated here as 0.33. And they did something else. They calculated the heritability also of microbiome features. So in this case, microbial genes. They calculated, and the microbial bacteria. So microbiome features heritability went from 0.13 to 0.61. So there were quite a few features inside that had high heritability. And in the end, looking at all the heritabilities and their genetic correlations and with methane and stuff like that, they selected 30 microbial genes that were genetically correlated with methane. And they will then test them as criteria for breeding, for selection of animals for breeding. And so I really like this image. So on this axis here, we have the methane in grams per kilogram of dry matter intake. And here we have different intensities of selection. So the first one is if we chose 30% of the herd to breed, then 20%, 10, 5, and 1%. So we are increasing the intensity of selection when we move to the right side. And here we have the three strategies. So the first strategy that is here, this light blue color, is when we breed animals based only on the methane emissions, the measurement. The slightly darker blue one here in the middle is when we use only these 30 microbial genes that were selected, and we see that there is a little bit of improvement. So obviously in methane, we want to decrease it, so it's going down, which means that obviously with increased intensity, we are going to get a little bit more gain. And then in the green one is a strategy that uses the methane and the microbial genes at the same time to select the animals for breeding. And we can see that if we go for higher, highest intensity, we could decrease 4 grams per kilogram of dry matter intake per generation, which is obviously quite good. This is not measured yet. This is just simulations to see what we could achieve. So I don't want to overpromise with this figure, but I think that it's quite a striking one, and it shows where we can go in the future. Okay, so take-home message is including microbiome information into the breeding indices leads to greater gains per generation. Leads, maybe I should say could lead, but I work in microbiome, so I do feel like it leads because it just, it gives us more information, so we have more to go on. Okay, so this one, again, the image seems a little bit complex, but it's very easy to understand. So in this work, so the previous work and all the actually most of the previous works that we've done are based on sampling the microbiome, sampling the rumen fluid at slaughter. And it's really good that we are able to sample at slaughter, and then we identify which microbial features are important for each little thing. But then for breeding, it doesn't really make sense because the animal is at slaughter, so it's dead. And we cannot really do anything after the animal is dead. So here we looked at the temporal stability of the microbiome. And so here it's again beef cattle, and we sampled the animals in 6 different time points. The 6th time point is slaughter, and the first one is 7 months before slaughter, so 1 year and a little bit of age. So this is just the experimental design, the time points that we sampled the rumen fluid. The sampling method is also a little bit different. So when the animal is alive, we put a tube down the throat and we sample the animal. At slaughter, we just open the rumen and we get the sample. And yet, when we look here at this, so in this case, I'm looking at two kinds of information. So here is microbial genera, so bacteria, archaea, protozoa, everything is here. Each dot is one animal, and each different color is a different time point. And we can see that everything is very cloudy. There is no structure, there is no clustering per time point. We cannot look at this plot and say there is an effect of time point. There isn't. That is what we can see from this image. In microbial genes, it's the same thing. So here is, it's just we are looking at microbial genes, and we can see again that this is very cloudy. They are all mixed, and we cannot say that one time point is different from the other. And then, so we did have not only based on this image, but based on a lot more analysis, but I think that this image shows that. So we already had the idea that the microbiome is temporally stable. But then is the microbiome relationship with traits temporally stable? And so then I went to check that part. And so what I did here was we took the microbiome of each time point and we used it to see how much it is associated with each different trait. And so we have, in this case here, we looked at feed conversion ratio using just the microbial taxa, so microbes. And the time point that explained the least explained 53% of the feed conversion ratio. And the time point, and this was time point 6. And the time point that explained the most was the time point 2, and it explained 74% of the feed conversion ratio. What I wanted to show with this is that the minimum, that is like the darkest color that goes up until here, and the maximum, they are not very far away from each other. We are always going to be very close. So this means that if I use any of these time points, I'm going to be between the 74% and the 53%, which in terms of microbiome is really good. And so on and so forth for the others. So this is just a different plot. So the top ones are for the microbial taxa, so the bacteria, the archaea, protozoa, and fungi. And the bottom row is for genes. And it happens a little bit the same thing. If we are explaining more than 50% with the worst time point, then it's fine. And then I also did a little step further. So because now we could say, yes, you are explaining similar percentages of variation of each trait, but the microbes that are important for this explanation maybe are not the same between the different traits. But we also checked that, and everything went very well, and it went very smoothly, and because the microbes were very stable over time as well. This paper is also published, and I think it's publicly available. So it's if you want more details, it also exists, and also obviously questions here if you want. So the take-home message of this one is that microbiome is temporally stable. So if we want to breed, we don't have to wait until slaughter to sample the animal and then do nothing with the information. Obviously, we are going to want to sample as early as possible, and the earlier we sample, the earlier we have the information, we can integrate it into the breeding indices and then use this information and breed the best animals. Okay, now we got to the sheep. So this is the last part. So right now we are doing just a preliminary analysis. We just started this. So I don't want to promise that you are going to see something revolutionary yet. So here we did the 250, we had data on 250 animals, and we had the methane measured in the portable accumulation chambers. And based on this data, after cleaning, we had 309 microbial genera, so 309 different genera of microbes. And we did this plot here. So this plot is just, I took the information from the microbiome and I put it in an image. Each dot is one animal, and very clearly we see there are two distinct clusters. And this was quite good because I didn't have yet information about anything else yet. I just had the microbiome, so I decided to do this, and I was very curious about what could be driving this separation between these two clusters. So one thing that we can do with this is that we can color the points based on whatever factor we want to color it. So in the first instance, there is a slight difference here because in the second one I only used 206 animals, but that's for like statistical reasons. It doesn't really it's more technical reasons than any other thing. But you can see that this is the same plot because there's like a ball here and then it stretches a little bit, and then here the ball and then it stretches a little bit there, and then here again a little bit more like elongated group, which is this one here. But basically it is the same plot. And what we can see is that when we color it by gender or sex, it matches perfectly. So we have a perfect separation. But there was a trick here. The separation between between sexes, matches perfectly with the separation based on age. And so right now, what we know that there are two very different groups, but we do not know what is driving this difference. We can have like inklings. It would be more natural to expect a difference based on age than based on gender. But what we know right now, based on the crude information that we look at the moment, is that we don't know yet which factor is leading this difference, but there is a very big difference. I also did the coloring, but based on breed. I just kept this one just as a reminder of what is from before. Here we can see that the breeds, again, it's very cloudy. We see the same clustering pattern because there is still the sex information is still here, or the sex/age information is still here. But in terms of breeds, everything is very cloudy and very mixed. So there's no real clustering, there is no real pattern. So most likely the clustering is not due to the breed. And then I did a little bit more just to compare the male younger group with the female older group. So one of the things that we look at in the microbiome is richness. So the richness is just the number of different species that we have in the microbiome. And the richness is higher in the male younger group than in the female group. Okay, continuing. The Shannon index. Shannon index shows us if there are some species that are very dominant and then the others are very low abundant, or if they are like very evenly distributed within the sample. And what we saw is that the Shannon index was lower in the female older group and higher in the male, very slightly higher in the male group. What this means is that there is a little bit more even distribution of the bacteria that are there, and the microbes that are there are a little bit more evenly distributed. So in terms of these things here have been observed in other animals and have been observed in cattle, and most likely it's because when the animal is younger, it gets colonized. A lot of different things are coming into the animal, through the mouth, and there's a lot of confusion as to who is going to be established in the rumen. And during the younger moments of the animal's life, there is a little bit more diversity and more things entering. And then at some point it stabilizes. And again, like considering also the weaning and the changes in diet and stuff like that. But there is a moment when the animals are adults, that they actually get a characteristic microbiome, a microbiome that is going to characterize them and still be variable enough that we can distinguish animals very, very easily. Then, based on a certain other index that is called Bray-Curtis, so we calculate the difference between one animal and each one of the other animals, and we construct a matrix of distances between different animals, and we color this by whatever factor we want. In this case, it was sex, and when we colored by sex, we saw that the males are much more together, and so they are much more similar to each other than the males. The males are much more closer together than the females. And so, again, we don't know if this is because of the sex or because of the age. It's just, right now, we are just observing, and this is what we see. And I don't want to make too many assumptions of why is it that we are seeing these kind of things. In terms of breed, again, we cannot really say that there is something in terms of distances between the animals that is driven by the breed, which is good in terms of analysis. So, take-home message for the sheep is that there is a distinction between the male younger and the female older groups, but we need a lot more analysis. And I think that's me. Thank you for the attention and any questions. And I hope I didn't go too fast.
Thank you, Joana. That's really useful. I love the way you say that this is very straightforward. Whereas I suspect most people are thinking, oh, God.
Yeah, I tried my best. I tried my best to make it a little bit, easy.
Yeah. We've got a question up from John over in Ireland again, which is asking really, could a probiotic type dose be used to seed the microbiome with the beneficial organism? So in other words, can you influence that microbiome makeup by actually putting probiotic dose with specific species in, I suppose?
Short answer, yes, you can. But there is a very big but. The microbiome has a tendency to maintain itself and to come back to what it was before the probiotic. So you are either going to keep doing it, and the animal, because the animal, as soon as you stop it, or not as soon as you stop it, but very, very shortly after you stop the probiotic or the prebiotic or whatever it is that you are doing, the animal will revert back. And this is one of the things that we think is related to the host influence over its own microbiome. So probiotics are really good. I feel like if you, for example, in this case, that if you give some antibiotics to the animals, if you are giving probiotics for the animal to recover faster from the antibiotics, the impact on the microbiome, then that would be really good. And if the probiotic is really cheap and accessible, great. But it's not permanent, and it's not going to go into the next generation. So, yeah, it's a yes, but.
Yes, but short-lived and needs to be repeated. Yeah. No, that's very clear. And obviously, our interest in all of this is very much along the lines of if you remember back to some of Joanna's slides there, if we can actually incorporate the microbiome species alongside some of the other traits we're measuring, then we can actually make much quicker progress.
Yes.
So yeah, that's good. So John's just coming back there asking, if the probiotic is not truly beneficial, is the diet a bigger influencer, which I think you've already said to some degree?
Yes.
Yeah.
So we can use the diet to influence what is going to happen in the microbiome. Yes.
Thank you. And then on the back of that, we've got Bill coming in saying, how do methane suppressing supplements affect the microbiome, which is probably very topical in the whole that happened just before Christmas. So maybe can you just explain how those additives and stuff actually have that influence?
So it depends on what will happen with this suppression. So there are ways of suppressing without actually, I don't know if suppressing is the word. There are strategies to increase the bacteria that are using hydrogen. And if some other bacteria are using the hydrogen, then the archaea cannot use the hydrogen. And therefore, they enter in competition. And we have a slightly different volatile fatty acid production, but we don't have so much methane. So there could be advantages in doing this. Again, it's usually short-lived because the community, although for us in the naked eye, it still varies a lot and it's still a lot of information. In terms of a biological system, it works in a very strict way. And so there are ways that we can act, but it has its own life happening. Anytime that we close one hole, another one is going to open. And because it is very complex. And we don't know yet. There's a lot of things that we still have to study about it. So I cannot say that, okay, if we suppress the methane, this is going to happen. It depends on how we do it. There are ways of just exhaling the hydrogen and nothing happens to the other communities. But again, we need a lot more studies, I would say, and to be a lot more specific about which kind of suppressor we are talking about.
And I'm sure we will see lots of these appear, including vaccines and all sorts of others over the next decade, I suppose. You know, our interest very much in all of this is to keep it as natural as possible, to select those animals that have that microflora and fauna that benefits us and it, because it's sustainable and we can actually breed for it, hopefully. So that's very much where we're heading with it, we hope. And as I'm sure most of you will realize, Joana is a pretty important cog in all of that, because we would be absolutely lost with some of this without it. So we're very grateful, Joana, for you making it seem quite simple in your mind, whereas Janet and I are running very quickly to sort of make sense of a lot of this stuff. But yeah, we have lots more samples on their way to a lab currently, and we will be sampling another 300 sheep in a few months now as well. So we'll build this data set. We'll get some more information on sheep, which will hopefully then enable Joana, from her expertise on the cattle side and the SRUC team, to actually really help us disentangle some of this, which will hopefully then apply itself to the wider industry. So thank you, Joana. And based on that, I suppose, Janet, it would be appropriate for you to talk about how on earth are we actually going to try and convey some of this work to the wider industry, because having these secrets and these evolvements is no good unless people know about them and can use them. So over to Janet for the last part of this, really.
Okay, can you see that?
Yes.
Yeah, so it's a really important part of the project, is just looking at how we can change methane, understanding all those complex interactions and why they're happening. But as Dewi says, when it comes back down to farm level, we've got to look at it in a bigger way. So part of the project is looking at in terms of, okay, if we make genetic changes to our animals in terms of breeding animals that will have a lower carbon footprint naturally, but how does that sit within the whole farm system? How is it going to affect the whole farm carbon footprint? And we know that genetics can intrinsically change the animal to produce in a different way, but it also can allow you to change the system in a different way, which will have knock-ons for the farm carbon footprint. So as part of Breed for Change, we're also trying to look at that. And we're basing it on data that's been collected in the Pilgrims supply chain. You might have seen Pilgrims are a partner in the project and a very valuable partner. So they've collected data from 62 of their lamb supplier farms. And we're going to do some modelling work with Harper Adams University to look at those changes. So if we make a genetic change, how does that change farm carbon footprint? How maybe does it allow you to change your system in a way that changes farm carbon footprint? So understanding those higher level interactions. So that will help us to understand that and how people could integrate livestock in different ways to improve the carbon footprint of the farm. It will also help us to refine some of the metrics we're using to measure the carbon footprint. And ideally, we'd also like to be able to provide data for some of the carbon calculators, because we will have a whole wealth of data in this area. And often in terms of their modelling in sheep flocks, they have fairly limited data to work from. So we're hoping to be able to work with them and provide some of the data for that. So we get more accurate carbon footprints for flocks. So we're looking at it in that holistic way. And we're also, as Dewi said, it's all very well us doing this, and we're learning a lot and a lot of very valuable information. But how can the rest of the industry benefit from that? So there's another piece of work as part of the project that focuses on sharing what we learn. So what's the best way of presenting some of this very complex breeding information on methane reductions for sheep breeders and sheep farmers, so that they can take advantage of it and integrate it into their own systems, their own flocks? And how can we use what we learn to reduce carbon footprint throughout the industry, not just in the flocks involved in the project? And by doing that through identifying the best breeding strategies to supply breeding sheep to help reduce carbon footprints. So the UK Agritech Centres and NSA have been doing a lot of work with us in terms of trying to just understand better ways of presenting this sort of complex information for sheep breeders and farmers. And we'll be developing that and rolling that out over the remainder of the project. So in summary, really, it's a big project with a lot of very complex parts to it, but trying to cover the whole area in a kind of holistic way to find the best way to breed ewes with a lower carbon footprint. We're focusing it on our populations, which are already very well recorded on grass and forage-based systems. So the solutions are going to be directly relevant to those sorts of systems, but probably applicable in a much wider population and a wider range of systems as well. And a lot of what we're doing is collecting data to develop these new breeding tools, these new exciting areas like the microbiome, and using that to understand what we're doing and identify the best breeding strategies. But we're also very committed to sharing the findings throughout the industry so other people can benefit from what we're doing, because it's a big problem. Individually, we're not going to tackle it, but if together we can all make good progress on it. So really, that's all I've got to say on it. Thank you very much for listening. It's an exciting area, and watch this space for more news and more updates. So I'll hand back to you at that stage. Dewi?
Thank you, Janet. Go back in here. Yeah, thank you for that for us. We did actually have a couple of questions which I missed just on the transition. So we've got a couple of microbiome-based questions for Joana, which if she's happy to cover with us, that'll be great. So Emily came in with a question, obviously mind blown, as she puts in her message. How much is the microbiome influenced by the environment, e.g., for example, diet, quality, the quantity, minerals, health, and have we missed the influence, anything and have we missed the influence of microbiome and some of the production traits we've already been selecting for? Does that make sense for you, Joana?
Yes, it does. It's a really good question and a really complex one. So, okay, how much is the microbiome influenced by the environment? A lot. We've been seeing differences. One of the challenges that we have in analyzing microbiome samples that come from different farms is the fact that they come from different farms. Immediately we can see it in the plot. So it's immediately there are different clusters. So to analyze this, we usually have to correct the data and try to remove from the data set the effect of farm. And many times are farms that are very close to each other. Many times is the same farm, but in different days. And we can see immediately difference. We can see differences within hours because of the feeding schedules and stuff like that. So the goal to overcome those in terms of analysis is get more and more samples and more and more samples. And at some point we are going to see what is the stable thing, what is the thing that is always there, what are the main actors in the methane or whatever production trait. In regards to the other part, we can, yes, influence the microbiome through the diet, through anything related to management. Yes, I'm guessing season will have also an effect. It's the season of birth and all of these things. I think that these have an effect, but I'm hoping that the effect of other things will be greater than this one. And we will capture them, and especially if we do the statistics properly and if we do the analysis properly, we are going to be able to get to the bottom and get to the information that is actually going to be useful in terms of the breeding. So it's good that that the microbiome is influenced by these factors, because it gives us information about these factors. But on the other hand, when we are trying to go towards an objective, we need to get rid of a little bit of this variation and then move on. In terms of health, actually, we have a work coming out, I hope soon, in regards to parasites, nematodes, helminths, Ostertage as well in dairy cattle, actually. We do see differences between the animals that were vaccinated, animals that were infected, animals that were never infected. We do see this kind of difference. So it is something that we are starting to look at. And it's true. If the microbiome is under the host influence, under the host genetic influence, and we have been selecting animals for millennia, we are doing selection of microbiome, unintended selection. But hopefully now that we have the knowledge, and we'll correct anything that needs correcting as well. So hopefully nothing bad has been done until now. And I think that now we are also like fine-tuning a little bit more our breeding and our breeding objectives. So it's a little bit a more precise kind of strategy that we have now in relation to what we did in the past. I hope I gave an answer to all the points in that question. Okay.
You know, I think so. Emily is saying it's a missing piece of the jigsaw that we're finding.
Yes, exactly. And it's a huge piece, and I think that it's great that we found it now.
Yeah. There was a question from Michael
yes.
in there. Could we be influencing the microbiome at birth, or could that be important?
Again, loaded question. So there are some researchers that think that, or they defend the idea that there is a window very close to birth that we can manipulate the microbiome in that moment, and then those changes are going to be kept over the lifetime. So we know for now, for sure, that, for example, a cesarean birth is very different from a natural birth. The type of colonization is very different. Even in humans, they have shown this. So for sure, the birth is one of the most impactful moments. And they also checked which was the main source of this colonization, and between the environment, between the different farms, different barns, if it's outside. And actually the main one is the vaginal microbiome. That is the main source and the main influencer. So obviously when we do the cesarean versus a natural birth, immediately it's completely different, also because of the fact that the natural birth is a much more extended event, and it's a little bit more progressive, and the other one is a little bit more radical and more different. Everyone knows what are the differences. So yes, this is a very, very, and there is this theory. For now, it's a theory that there is a window of opportunity when they are very young. Now, if this is true, we don't know yet, but it could be. It could be because it's like any population when we go to a territory, right? The bacteria will go there, and the first ones that get there, they start getting established. And if they do well there, then they are not going to allow any other bacteria to come into the environment. So they are going to be a little bit protective of that environment. So yes, for sure, the birth is extremely important.
So a very appropriate question, Michael, and answer obviously is yes, although we're not quite sure to what degree and how yet. But yeah, as we are exploring this this whole area, I think we will be turning over quite a lot of stones and finding lots of things. So yes. And we will be very engaged and invested in doing this. So yeah, we'll keep you up to date with it all. Michael has also shared a link in there, I think, of methane measurements being taken, which on the BBC stuff, and there's some various videos as well on the Breed to Change website and links if anybody wants to get hold of them there. I am going to draw the evening to a close. We've just turned 9 o'clock, so I'm conscious of Joanna needing to go to bed, and I'm sure you've also got things you've got on. So thank you again for joining us. We've been really, really pleased to have so many people come on board. A recording of this will be sent to everybody that have registered, and it will be available probably on the Innovus YouTube channel in due course as well. Our next one is in a fortnight's time, which we'll be talking sheep in arable rotations and specifically partnerships with our own breeding partner, Matt Harding, and one of the farmers he works very closely with, and the crucial things really that are needed for making those systems work in a true partnership form. So that'll be a fortnight tonight on the 21st, and we hope to see lots of you there again. But thank you, Joanna. Thank you, Janet. And
thank you.
Thank you all
Thanks
for
for
joining.
having me.
And take care. Good evening.
Thank
Bye-bye.
you. Bye-bye.
Bye-bye.